So today I would like to share the mutational spectrum of GNE myopathy cohort in China. I intended to share some preliminary natural history data, but the patient enrollment is not as good as I expected. So probably I can share with you next year. Okay, so you know all about GNE myopathy, and which is characterized by rimmed vacuoles on the muscle pathology. And also, it is the most common distal myopathy in China. It comprises almost half of our distal myopathies in our single center cohort. Okay, so actually, we started muscle pathology from 2005, and after years and years, we actually have diagnosed more than 100 GNE myopathy patients, and most of them have a muscle specimen in our biobank. Okay, so GNE mutation, most of them are missense mutations, in which the hotspots are marked here and over both the epimerase domain and the MANAC kinase domain. And we also found that previously there are some special mutations can also lead to like some synonymous mutation near the boundary of exon intron boundary. They can alert and they can alter the mRNA splicing. And also there are rearrangement hotspots encompassing the 5-UTR due to the high density of ALU sequence in the introns. However, what's bewildering is that in our cohort, there are still remain 11% of patients who are who have only monoallic gene mutations. So there comes this index patient who is a 30-year-old young man. He complained of distal weakness at age 28, and it progressed quite fast. And the EMG showed primarily it shows a neurogenic change, and he was diagnosed with Charcot-Marie-Tooth. But what is notable is that he have very early epistaxis since 3 years old, and then he developed thrombocytopenia and was diagnosed as idiopathic thrombocytopenia purpura, and got the splenotomy. Okay, so we can see that he still has a very low platelet count and elevated CK. And what we can see is that there is very prominent distal muscle weakness. But on the muscle MRI, we could see that actually this patient is peculiar because the quadriceps is spared, even in the very severe involvement of the legs. And the muscle biopsy on the TA showed rimmed vacuoles and very severe myopathic change. And also there is P62 deposition. So we did the whole exome sequencing for this patient and found one nonsense mutation, and the other is synonymous mutation. So that this patient have a GNE myopathy. We further performed mRNA analysis, and we don't find any aberrant splicing involved by this synonymous mutation. And also we performed exon 2 qPCR, and we don't find any copy number variation encompassing exon 2. So when we look back to the clinical history, we can see that the distal myopathy has many related genes. It can be autosomal dominant or recessive patterns. And some of them actually can have the pathology as rim vacuoles, but only GNE have been reported to be related to thrombocytopenia. So we decided to run a second test, a long-read sequencing for this case, and it called out a deep intronic variant. So this variant actually is, we did parental analysis and found that it is in trans with the other nonsense mutation. So the BDPG, that is some software, and also Splice AI predicted aberrant splicing of this deep intronic variation. And also, we performed qPCR using muscle samples from this patient, and we do see an extra band with 100 and 231 BP insertion. And so actually it will create a pseudo exome insertion in intron 5, and which was predicted to lead to a truncated protein. So then we further analyzed the other patients with monoallelic mutations in our cohort, and we did find 8 more patients with this DIV. Among them, 3 also have a thrombocytopenia. And after we did, some of the patients actually they have all of these variants. If we can have the pedigree analysis, we can see that they are inherited from their parents, and by haplotype interfering, we can see that these DIV are on a common haplotype. And also, this was further verified in other patients, both by qPCR and in some patients with the muscle sample adequate. We performed RNA-seq and see a very small amount of the transcripts have this pseudo exome. So what make us not fully understand is that previously, as we know, it is reported that there is a rule for those GNE myopathy with thrombocytopenia. That is, most of the patients, they will have at least one mutation on the manic kinase domain to get thrombocytopenia. But in our 3 cases, alongside the besides the DIV mutation, which will cause aberrant splicing here, most of the other mutation all lie in the epimerase domain. So that's very peculiar for these 3 cases. And then we also want to know if this DIV mutation is underestimated in the general GNE cohort. So we actually called on all the neuromuscular centers around China and asked them to submit unpublished cases with or without monoallelic mutation. And finally, we actually collected 113 new cases unpublished. So in this cohort, we actually find 2 more DIV patients with DIV mutation. And so this leads to this DIV actually ranked the top 10 variants in our cohort. And the most frequent mutation in Chinese population, as previously reported, is still the D207V mutation, which comprise about 1/4 of the whole alleles. So the other thing that amazes us is that, just like the NIH group just reported, we also find many, about 23 novel mutations along the GNE gene, which has not been included in the database. So the pathogenicity of these novel mutations remains to be determined. And also, in this study, we find one more patient with the allo-mediated deletion of exon 2 to exon 9. And also, we verified previously reported cases. We found that using long-read sequencing, there is an insertion of 639 base pair of unknown origin. So we also performed a genotype-phenotype correlation by retrospectively ask the patients to recall their history and the year they lose their ambulation. And so D207V is the most common mutation in Chinese cohort. And the Kaplan-Meier survival analysis revealed that the patients with D207 actually is the milder phenotype. Those patients harboring at least one copy of 207, actually they will remain ambulant even in their late years, while the DIV patients and the other patients, the natural history is quite the same with the other countries. So that's my conclusion, is that GNE myopathy is predominant in Chinese distal myopathy cohort, and the deep intronic variant is related to both GNE myopathy and thrombocytopenia, which is very important because this mutation will not routinely be checked in our clinical consolidation, and some of the patients, actually, I dig those patients out from those peripheral neuropathy cohort. And the carriers of D207 mutation progress more slowly than those other patients. So this also leads to the caution that when we perform natural history analysis and also clinical trials in Chinese population, we need to do the genotype stratification when we analyze the outcome results. And so now we are just at the start of build our own Chinese GNE myopathy registry, and I really hope to get help and advice from all of you who are very experienced in this field. And I want to welcome you if you have chance to visit our hospital based in Shanghai. This is the very old building, the first building of our hospital. And this is the downtown campus, which is very crowded now. And now we also have a West Campus, which is the localization of National Center for Neurological Disorders. So that's all. And thank you for your attention.